Good morning, class. I hope you are doing well wherever you are watching us from. My name is John, your online biology teacher, and today we are going to look about the homeostatic roles of the liver, and we will begin with regulation of blood sugar level, deamination, and detoxification. All right. I've said today we are looking about the homeostatic role of the liver, and we are going to look about the regulation of blood sugar level, which is glucose, the deamination, which is breakdown of excess amino acid, and detoxification, which is the breakdown of toxic substances to less toxic forms. So in regulation of blood sugar level, you have to know that the regulation of blood sugar level, it is made possible by two hormones, the insulin and the glucagon, which are released by the liver cells. And these two hormones, insulin and glucagon, they are antagonistic. They are antagonistic in that they work in the opposite direction. And the one as one drops the blood normal to normal, the other one raises it from below normal to the normal range. All right. So when the blood sugar level rises above the normal range, the liver cells are stimulated to produce insulin. I have said that when the blood sugar level raises above normal, the liver cells are stimulated to produce insulin. So in this, you can see that now that the blood sugar level is above the normal range. So the liver cells are now stimulated to produce insulin. So insulin drops the blood sugar level to normal range by stimulating the conversion of excess glucose to produce energy. That is number one. It stimulates the conversion of excess glucose to produce energy. The other one, it stimulates the conversion of excess glucose to glycogen for storage. And number two, it stimulates the conversion of excess glucose to glycogen. Number three, it stimulates the conversion of excess glucose to fats for storage. Are we together up to that point? We have said that when the blood sugar level rises above normal range, the liver cells are stimulated to produce enzyme known as an hormone known as insulin. So insulin drops down or brings about blood sugar level to normal range by stimulating the conversion of excess glucose to glycogen for storage. It stimulates the conversion of excess glucose to fats for storage. It stimulates the breakdown, or it increases the oxidation of excess glucose to produce energy, and it inhibits the conversion of glycogen to glucose. Are we together up to that point? I've said that the insulin also inhibits the conversion of stored glycogen to glucose. Let's now check on it when the blood sugar level drops below the normal range. So when the blood sugar level drops to normal range, the liver cells are stimulated to produce glucagon. Glucagon brings about the blood sugar level to normal by blood sugar level to normal range by stimulating the conversion of stored fats to glucose. That is number one. It stimulates the conversion of stored fats to produce glucose. It stimulates the conversion of stored fats to glucose. It stimulates the conversion of stored glycogen to glucose, and it lowers the oxidation of glucose to produce energy. So it lowers or it decreases the oxidation of glucose to produce energy. And in that case, we'll find that the blood sugar level is taken control of. All right, so that's all about the regulation of blood sugar level, which is glucose. Now let's look about deamination. All right, let's look about deamination. So deamination is the process by which the excess amino acid are eliminated by the liver cells. All right, so deamination is the process by which excess amino acid are eliminated by the liver cells. All right, so amino group which is present in amino acid is used to form ammonia. It is used to form ammonia. That amino group, it combines with hydrogen to form ammonia. Ammonia is now taken to the enzyme cycle. Ammonia is now taken to the enzyme cycle, where a series of reactions takes place to produce urea. We have said that deamination is the process by which excess amino acid are eliminated by the liver cells. An amino group which is present in excess amino acid combines with hydrogen to form ammonia. And we have said that ammonia is taken into the enzyme cycle, where a series of reactions takes place to form urea, which is a less toxic substance than ammonia. And enzyme agenase speeds up the reactions that forms urea in the enzyme cycle. You can be asked which enzyme is involved in the formation of urea in the enzyme cycle. It is enzyme agenase. So we have said that deamination is the process by which excess amino acid are eliminated by the liver cells. And we have said that amino group which is present in the amino acid combines with hydrogen to form ammonia. And ammonia is taken into the enzyme cycle, where a series of reactions takes place to produce urea, which is a less toxic substance than ammonia. And enzyme agenase speeds up the reaction that forms urea. Urea is later on excreted through the kidney cells, and the other remaining organic substances are converted into glucose. Other remaining organic substances are converted into glucose. All right, so that is all about deamination. Let's now talk about detoxification. We have said that detoxification is the process by which toxic substances are converted into less toxic substances. Are we together up to that point? We have said that in detoxification is the process by which toxic substances are broken down into less toxic substances by the liver cells. All right? Due to many reactions in the body, you'll find that there will be hydrogen peroxide will be formed. And hydrogen peroxide is a very toxic substance. So the liver cells are stimulated and they therefore convert this toxic substance, hydrogen peroxide, into oxygen and water. Are you alright? So this, in detoxification, we've said it is the process by which toxic substances are converted into less toxic substances by the liver cells. And detoxification is normally, in practicals, is normally tested when you are given a potato tuber. You have been given a potato tuber, you have crushed it, and you have been instructed to add hydrogen peroxide. All right? Yeah. So this is mostly done when you have been given that potato tuber, you have crushed it, and you have added hydrogen peroxide, and you'll normally see bubbles. Right? You normally see bubbles, and sometimes you are told to test about the gas produced. All right? So that gas that is produced when hydrogen peroxide is added to crushed potato tuber, it is oxygen. And there is an enzyme which speeds up this reaction, the conversion of the breakdown of hydrogen peroxide, and it's known as enzyme catalase. All right? Yeah, it's known as enzyme catalase. It is the one that speeds the reaction, or it is the one that speeds the breakdown of hydrogen peroxide to oxygen and water. All right? Thank you for watching up to that end. I'll give you an assignment to check the rest of the homeostatic role of the liver. We have hemoglobin regulation, we have thermoregulation. Yeah, so you just go and look about them, and I will see you in the next class. Thank you so much for watching, and kindly you can like, you can comment, you can subscribe if you have not subscribed, and you can type below, or you can comment below which essay you'd want us to look next. All right? Thank you so much.